All right, thanks everybody for putting up with another talk by me. So since we have not that much time, I'm thinking also maybe some of the stuff I can talk about tomorrow in the morning. Yeah, so we'll get to what I get to. I might also ask the audience for some feedback about maybe what sections I can skip over if everyone basically agrees. This is the outline. What I want to start with is some of the goals and rationale behind this notion of automated landmarking, and I think sections that maybe I could skip over is a section on limitations of traditional landmarks. Traditional in the sense that they require a user to actively look at and place landmarks. I think we could probably all agree that if there was a good method to automatically place landmarks, we would be fine not doing it ourselves. So I might skip over that part. I'll give a definition of automated correspondence, and then talk about the implementation that we have developed called Auto3D-GM. Many of you have probably heard about it and frustrated with it, and so I hope to present what I think are some good updates to it, along the lines of, Mike, what you were talking about, computational improvements that make it a lot faster. And then if I have time, and this might be what I'll talk about tomorrow if I don't have time, going further, some other things that we can do. But I think it's important to sort of to step back and think about like what the goals of at least in my understanding, morphometrics generally is. And I think really what we're looking for is to achieve data-driven understanding of the pattern of micro and macro evolution that describes the anatomical diversity of life. I feel like that is ultimately what we want. And I think if that's what we want, what that requires is to be data-driven, quantitative approaches capable of meaningfully aligning and or comparing homologous structures, and to use the classical definition, the same organ in different animals under every variety of form and function. And the rationale there is that I think without such a framework, comparative morphology will remain sort of arbitrarily limited in its ability to quantitatively describe morphological and potentially evolutionary continuity uniting different individuals and species, according to disparity in some animals not having certain parts and sort of thing. So I think again there's a variety of limitations to traditional landmarks. That's not to be a criticism to studies or a criticism of the reliability of results from studies using traditional landmarks, just that they have a number of limitations that we would love to overcome. One of the classic problems is that we know if we have inadequate coverage, we're going to have an unstable characterization of shape. And there's some neat demonstrations of this by Aki Watanabe a few years ago. The other thing is that the data to represent shape have to be chosen a priori, at least a couple of features if we're going to use semi-landmarks. And this is, if you look in the textbooks, as a general rule, landmarks should be chosen so you can quantify any differences that you can see. So that's coming from a point of subjective, I think I already know what the important features are. And of course, in practice, when we start to choose landmarks, well, that pointy feature looks like a good place for one, but then all of a sudden we get to a specimen and reasonable people can start to disagree about what the corresponding point should be there. And this criterion of homology is technically not even defined on within homologous structures, and of course it can be difficult to substantiate in practice. Let's see here. If you're trying to sort of force a guess there, that's going to lead to inflated intra and inter-observer error on placing those landmarks. And of course, we can't use traditional landmarks, at least not fixed landmarks, for observing topology changes. So we can't observe the loss of a paraconid based on landmark positions that require identifying the paraconid cusp. This is the paraconid cusp, that lower molar tooth. And if we're dealing with samples that are really different, we're going to have low amounts of coverage, which again leads to instability of shape characterization. And of course, you might have structures that where you know they're the same structure, but there's really no way to easily identify meaningful correspondences. So maybe automated correspondence determinations can get around this to some degree. So to be clear, what I mean by automated correspondence is that we can come up with a biologically, in some sense, loosely defined, biologically meaningful, possibly homologous points are identified between pairs of digitized structures without human intervention, except possibly through manipulation of algorithm parameters or or model parameters, depending on how we're doing this. So that's what I'm going to talk about. Let's see. So there, of course, people have been working on automated approaches for a long time. There's a lot of good ones in 2D analysis, but it gets more challenging when we come to 3D. And a lot of them, whether they're 2D or 3D, still require an observer to apply some sort of initial guess about correspondence. If you're doing, and when you think about it, when you're doing 2D automated landmark discovery based on photographs, there actually has been user input at the stage of photographing the element. Someone has made a decision about what the equivalent orientation is for that photograph. So it's not really automated because someone just decided the direction of dimension reduction. I'm just going to go through there. Some of the ones that you see published in the literature in in medical imaging or neuroimaging are working on samples with narrow ranges of variation relative to this goal of identifying correspondence among sort of the diversity of mammals or the diversity of vertebrates. And beyond everything else, they can be really computationally prohibitive. And that's probably the biggest hurdle, is like finding a way to get good alignments and registrations that aren't too computationally prohibitive. So letting all those, the alignment and the registration among digital shapes is a computationally difficult solution. There's sort of infinitely many ways to register two shapes to each other. So to find the optimal mapping is difficult. So one way that we started this process a few years ago with Sean and other members of that research group is to actually utilize procrustes to find the best candidate among sort of a reduced space of potential mappings. So the way we approached that, we had to reduce that initial space of original mappings by doing a conformal flattening of the 3D samples of interest by using the deformation as a function, identifying just a couple of candidate points to map, and then based on a few candidate points, defining a couple of different mappings, and then amongst those candidate mappings in this conformal space, choosing amongst them by expanding those registrations back out into 3D and using the minimum procrustes distance to go with to define as the correct mapping. So anyway, with that approach, we basically got plausible correspondence maps, ones that sort of looked pretty good, and that also had a distance matrix that did a pretty good job at classifying things according to species. So we compared sort of computer correspondences to landmark data sets collected by human observers in their ability to classify things correctly by species. And we did it on 3 different samples: teeth, radius, and metatarsals. But this still had some limitations. These mappings wouldn't really be usable in downstream analysis in something like geomorph, for example, because the mappings were non-transitive. So they were all just pairwise. So there wasn't any sense of, you know, a point that could be recognized as a sort of a unitary feature point along, you know, in the whole sample. In other words, the mappings are non-transitive. So you could, if you started on one surface and mapped that point to 2 others, and then transpose that onto another surface, you'd end up in a different place. And that's not what we need for classic geometric morphometric analyses. And so Auto3D tried to solve some of those problems with some more simplified approaches. And it basically relies on the idea that if you have 2 objects that are really similar, it's actually pretty easy to align, at least align them. That is, you know, you can there's a very like the best alignment will be have a much smaller Procrustes distance than the bad alignments. When you start to get to very different objects, you can't rely. All of the distances tend to be bigger. So what we essentially do, I'll just show the approach here. So we start by resampling to a set number of what we call pseudo landmarks, so evenly spread points over the surface, and then we compute the principal axes of each model or object in the sample, and those first 3 principal axes are our initial test alignments. So we have essentially 8 initial test alignments, and so there's some assumption about, you know, how much variability is going to be in the sample, but that gives us a starting point for essentially an iterative closest points approach to finding what we'll call our best alignment based on evenly spread landmarks. So it gives us some, you know, allows us to do something like that. And then we do that for all pairs, and actually then just determine a minimum, just compute the minimum spanning tree between those, and propagate the correspondences through that minimum spanning tree. In other words, we don't bother actually trying to compare the really different shapes to each other. We don't try to directly determine the alignment between shapes that have really high Procrustes distances in their pairwise mapping. And what that means is, you know, if we start off, if we're just going through running the algorithm, doing pairwise comparisons, you know, we get great alignments on shapes that are really similar to each other. At the first pass, we often have bad direct alignments between shapes that are, you know, that are different from each other. But then on the second pass, we revise the correspondences by only going through the path of this minimum spanning tree, and that allows us to fix those incorrect correspondences. So we can get pretty good alignments on, you know, on samples that have a lot of diversity to the shape. And so, you know, as an initial, as a test sample we did a long time ago, you know, a bunch of ankle bones. This is the automated result with 1,000 landmarks, and you know, we've got different groups of primates that are separating out, you know, and it looks pretty good. If we compare to a smaller number of user collected landmarks, you can see there's a lot of similarity in the aspects of shape that are being captured. So I think one and I guess just to make the point, one of the problems with this software is just that we didn't have good ways of accelerating the computation of everything. So if you were using 1,000 landmarks on 100 specimens, you know, on your local computer, it was probably going to take 2 weeks to run. It was ridiculously expensive. And then actually, if you're just using a small number of pseudo landmarks based on the way that landmarks are distributed, it doesn't work very well. So if you're doing with 100 pseudo landmarks, you're not going to get good shapes spaces if you're analyzing the pseudo landmarks directly. But now with the new implementation that we have, you know, an analysis that would have taken 30 hours will, at least if you have at least a couple cores on your laptop, most laptops have like 4 to 6 cores, you'll get it down to, you know, between 3 hours and 30 minutes. So we haven't gotten quite as much of a percentage reduction as you have gotten, Mike, with Geomorph, but it's livable, at least at this point, I think. I can talk about accessing the software and installing it tomorrow if people are interested. But one of the things about this, one reason people might not be happy with Auto 3D GM landmarks is, you know, we sort of expect these registrations to somehow reflect feature points, you know, or what we might call, you know, type 2 landmarks. So, you know, this is really what we probably, if we were landmarking manually, what we'd sort of expect. That's not the kind of correspondence you get with Auto 3D GM. You get something more like essentially just a grid applied over the surface. I don't know if that's a problem or not. I think a lot of people will say, well, this is obviously a problem because you're not mapping homologous features to homologous features. But I guess the question becomes, like, what does homology even mean within a single bone? I guess beyond all that, because I probably won't have time for it, what we can actually do with Auto 3D GM is just use the alignments as a starting point to simplify more feature-aware downstream registrations. So I don't know if I want to get myself in hot water by talking about the difference between homology and correspondence. I can, or I can also just like be done here for now and talk about this other stuff tomorrow. Yeah, maybe we can. Well, so maybe I'll just skip forward. So you know, I think you can ask the question. I'll take 5 more minutes. I'll just skip forward past this discussion about homology and correspondence. There are all kinds of things you can ask about it. I do think one point is that it would be nice if correspondence hypotheses could emerge from our analyses instead of be predetermined by it. That's one point. So this is the new software. And I think in terms, once you have a good alignment, I think one interesting thing you can do is essentially what we've tried to do is apply essentially a Gaussian process to find initial candidate points. So, you know, we sort of envision the surface as a function. We have sort of our Gaussian process where we sort of modeled the surface initially as a smooth surface. What we can do is we can sort of find points and pick points based on the maximized error of the initial surface function in predicting them. And we can sort of update that model of the surface, and then again apply that criterion, pick our next candidate point based on, again, it's how much error there is in prediction from by the model surface. And so what that allows us to actually do, if we apply that process to actually, picking candidate points on a surface, it's really interesting. So these are just 16 observer placed landmarks on a tube. They represent sort of classically recognized features and points. And then on this tooth here, the numbers represent, the sequence of points picked by Gaussian process landmarking. So, and what you'll notice is that most of them match really precisely features, quote unquote, subjectively chosen by a human observer. And so I think, and so what this does is gives us, instead of just like one or two points as based on maximum curvature to start the alignment and registration process, it gives us a whole bunch more. And so we can do this with different kinds of samples and essentially go through this process of then starting with these candidate Gaussian process landmark collections, and then winnowing those down and taking into account the whole sample so that we have sort of a smooth, as much as possible, a smooth mapping defining the whole sample. And then we have, again, these consistent, landmark-like points that are attached to features that we can then use essentially a thin plate spline to interpolate amongst for the whole sample. So I think there are other things we can do from there. Once you have a fully registered sample, obviously there's lots of interesting things you can do. So yeah, so I think I'll stop there.